Hello, this is Mr. Baker, and this is Unit 5.2, Photosynthesis. And we're going to be talking about how to describe photosynthesis as a chemical process. Your main idea is that during photosynthesis, these organisms are going to take light energy, they're going to trap it, they're going to use that light energy to convert it into chemical energy during this process called photosynthesis. There are two distinct stages: the light-dependent reactions, which require light, and they occur in the thylakoid membrane, and the light-independent reactions. They don't require light, but they still do happen during the day, and they occur in the stroma in the chloroplast. Photosynthesis is done by photoautotrophs, and there's your equation of carbon dioxide plus water yields glucose plus oxygen. And there are the two phases again: light-dependent reactions. The light energy is absorbed and converted into the energy molecule ATP, a fully charged battery, and the electron pirate in plants, NADPH. Light-independent reactions, energy from ATP and that electron pirate, NADPH, are going to be used to build up glucose. There is a video that you can watch on your own. There is the photosynthesis equation again, where carbon dioxide plus water with sunlight yields sugar plus oxygen. The light-dependent reactions have two parts, and these are very familiar because we just learned them last unit. There is an electron transport system that has same idea, different players, and chemiosmosis, which is essentially exactly the same for our purposes. Chemiosmosis is using ATP synthase and an H plus proton gradient to hook up ADP plus P to recharge the ATP molecule. Our battery charger is the molecule ATP synthase. ATP is a fully charged battery with 3 phosphates. ADP is a partially charged battery with 2 phosphates. Anytime that you have an electron transport system that's moving protons around, you will have an ATP synthase that will be close by to use that gradient to do this chemiosmosis. Where? In the context of plants and in the chloroplast, it occurs in this thylakoid membrane. And the thylakoid membrane is from last section. There's a video that we watched in class. And so the actual H plus is released inside the thylakoid. They want to cross the membrane. ATP synthase will let it cross, but at a cost. Each H plus cranks the ATP synthase once. You'd get to crank it 3 times, and that'll allow you to fully recharge 1 ATP. Here is the table that you filled out several times in class. And what is chemiosmosis? Where is the thylakoid membrane? Who is the battery charger ATP synthase? And how it uses that gradient. It wants to diffuse across, but it will charge it to cross. 1 proton is 1 crank, and 3 cranks get you 1 ATP. Here are the players in the electron transport system, and there is a bigger picture right here. Photosystem 2, you should know something about photosystem 2, more about that in a couple of slides. And then this guy was part of the homework at cytochrome B6F, then photosystem 1, and then it goes to, and we'll just call him F and R. You are not responsible to know those, but I just want to point out they're there. Photosystem 2 is a big protein that's found in the thylakoid membrane, and it is the big player, and you do need to know him. More in a moment. Another diagram of what's going on. Light energy is going to excite these electrons in photosystem 2, and that kicks off this process. These electrons want to move, but they need help. They need an input of energy, and that comes from light, or the photon in light. While you keep getting electrons taken away, you need to replace those electrons, and that's where water gets split. This was a fundamental change in the evolution of life on this planet, because once somebody, some life form, invented the ability to split water, it allowed this whole process of photosynthesis to occur. When water is split, oxygen is released, and in the context of evolution, when that oxygen is released as a waste product, set up our atmosphere that we now enjoy. That was a big deal. There's a picture showing you more of what's going on. And there's our players again. In years past, we made a bigger deal about those. So let's read through what's going on. And this is pretty much a summary of the video that we skipped earlier on. Light energy excites chlorophyll in the photosystem 2. These things shake, and they change that energy by what's called resonance all the way through to the middle to something called a reaction center. This is going to have enough energy to kick the electrons down to the next molecule, which is cytochrome B6F. That was that yellow dude. Water is then split to replace the electrons sent to cytochrome B6F. The electrons from cytochrome B6F are then moved to photosystem 1. They need another kick of energy to move them to the next molecule called FNR. And then they go from FNR to NADPH. And then NADPH, the plant electron pirate, is going to take those over to the Calvin cycle. And more about the Calvin cycle in a moment. What are you expected to know? The reactants for the light-dependent reaction, which are light and water. The products, which is oxygen, which is a waste product. NADPH, which is the plant electron pirate, which comes from the electron transport system. Fully charged batteries, the ATP, which comes from chemiosmosis. And where does this all occur? In the thylakoid membrane. The light-independent reactions are also called the Calvin cycle, and you can also refer to it as C3, and I'll explain that in a couple of slides as well. You can also refer to these, the Calvin cycles, the dark reactions, but that is a term that's out of favor. I'd prefer that you use light-independent reactions or use Calvin cycle. I prefer Calvin cycle. What goes in? ATP, NADPH, plus carbon dioxide. What comes out? Glucose. Where? Stroma. Here's a picture showing you that we've got the energy going in the form of the fully charged battery and the electrons from the electron pirate with some carbon dioxide that you pull out of the air into the Calvin cycle, and it gives you the sugar. That sugar is glucose. When the Calvin cycle pulls carbon dioxide out of the air, that's called fixation. It's taking carbon out of the air and putting into molecules that living things can use. Carbon fixation, and the enzyme that does that is called rubisco, which stands for ribulose bisphosphate carboxylase or something like that. You just, excuse me, you just need to know rubisco. And that's the one that does it. Rubisco takes carbon dioxide out of the air, uses the energy from ATP and NADPH to form glucose, and it does this during the Calvin cycle. There's a whole bunch of things going on here that we're not going to get lost in the details. Why is Calvin cycle also called C3? Because when plants fix carbon in a normal Calvin cycle, it's found in a 3-carbon molecule, hence carbon 3. Some plants tweak this and use a 4-carbon molecule, and they are called C4 plants. More about why this occurs later. I'll touch upon it a little bit, and it's in the next unit. So here's a summary of your Calvin cycle. In the summary, and this is optional, you don't have to write this down, I do expect you to know what goes in, what comes out, and where it occurs. And what better way to do that than with a table? Here's your table that you've written down a few times, and you can read that and memorize it, and that should help you. Cellular respiration and photosynthesis are the inverse of each other, whereas the products for one are the reactants for the other. So the products for photosynthesis are glucose and oxygen. Those are the reactants that go into respiration. So cellular respiration and photosynthesis recycle each other's products. And this is a good thing for life on the planet. Quiz 5.2 is what you need to study for, covering those topics. Hope this helped. Study hard, and we'll see you at school soon.